R.T. Newman is a mastermind, sitting class to pass the time. DNA, cell theory, write your notes and rhyme in mind. Our cells of DNA tip us on their information, cell to cell heredity. Now the cell theory in motion, yeah, DNA make it work, it's in the central dogma. Yeah, these strands duplicating, reproduction and mitosis and meiosis. Doesn't matter a song when you're thinking inheritance, it's the double strongs. Even natural selection's got DNA in its back. It markers of mutations get us on track. And if you're sitting there just thinking that you got all of this down, we are turning this shit, look the other way now. Here's a different read by C.H. Waddington. Yeah, it's called epigenetics, cause the genome it's above goes like hovering. Cells can change in this theory, yeah, nurture changes, nature phenotypes can vary. Oh, these changes are not random. What do? Can be environment induced, make direct changes to you, and they must be inherited. You pass on these things to your kids when these cells flick, it changes, no leave.
Time for us to list almost all the major ways epigenome mechanisms we know to this day. DNA methylation is first up to the plate. Make that base cell grow to a different cell type, Here's the big pick, it turns on and off genes. Are these changes passed down?
And Newman agrees.
The environment too can tune our different genes. Changes get inherited, mitosis, meiosis. Now it's time to zoom close in, here it is. Let's begin. We get so detailed that you'll pray for all the trees on the earth, man. We got DNA methyltransferase. These enzymes do DNA methylation. DNMT, slap on SCH3 cause they wanted it. DNMT, 3A and 3B put methyl de novo. DNMT, 1 is for the maintenance of all of this, so important. It's how cells keep changes that they collected. CpG sites is where all this usually occurs. If that site got methyl on it, expression is not there. It might prevent the binding of activator proteins, or it might mess with the histone. Brings me to another topic. The acetylation of histones can occur. Methyl brings the proteins like I bring home the smuckers. Yeah, chromatins on a spool, it is tightly wound around and round. Being so compact, yeah, transcription factors can be found. With this we can tell each and every tumor cell, apart from cells with normal methylation that go rather well. Now for the last of the ways, here we go. RNA interference with expression, no amigo. RNA induced silencing, RISC is its stand-in. First we start with primary transcript, folds into a double-stranded hairpin of dsRNA, which is pre-processed by dicer protein loaded in a gonad to form the miRISC. This interferes with translation, formation of heterochromatin, binds to a target with miRNA, and they can perform its gene regulation. Also helps formation of more complexes. This loop is silencing, positive.
To clarify, RISC can directly act on target DNA structure, where target DNA corresponds to the DNA that produces the target RNA, the RNA that matches RISC's miRNA strand. RISC causes the methylation of this DNA's histones, which then causes the formation of heterochromatin. In this condensed state, the genes on this DNA will be silenced. If any transcripts do manage to get produced, RISC helps turn them into even more RISC complexes, creating a positive silencing loop. Finally, note that since these changes are being made to the DNA, they're heritable. It's epigenetics. Can I end it?
Yes, you can.
Can I end it?
Yes, you can.
Can I end it?
Yes, you can.
Well, I'm gone.
Go on then.
Can I end it? We got a paper that explains what we now know. Condition mice to receive phenome. Shock smell condition to be sensitive. Extra neurons in the nose and brain live. But the shock is the next generations. Sensitive even though never smell the stuff. Also had the extra neural sensor so buff. And the reason is that epigenome is enough. Passing changes messing with the strand stuff. It's the nurture was rolled out by negative control corresponds to the mice. Propanol control show the smell specified.
Hold up. When mice were conditioned with propanol, the acetophenone responsive gene structure was just fine. So you see, it wasn't just the shock and stress. Not those variables. It was really that fruity almond cherry scent. They even look at a completely different olfactory gene up in there as a negative control.
Yeah, with the changes from the big guy. Check the sperm of the parent and the small fry. Cytosine gets converted with bisulfite. Unless it's methylated, then it stays laser guy. PCR determines where the changes lie. Amplified strands converted with bisulfite. Gene is less methylated, we say oh my. Learn trait passed down in